Good afternoon. I'm Anne Rosser and I'm giving this talk on behalf of Stem Cells for Huntington's Disease, SC4HD, and the EHDN Advanced Therapies Working Group. And the steering committee members for these groups are included in the list at the bottom of this slide. So I'm going to briefly review why we need a programme of work around clinical translation of cell therapies and what we've been doing in this area over the last couple of years. The underpinning concept of cell therapy is restoration of structure and function. Within the Huntington's community, we're familiar with the idea that the striatum, shown here as a schematic, is disproportionately affected in Huntington's, and that degeneration of the striatum begins many years before symptom onset. This relative focus of degeneration provides a target for cell therapy. And the purpose of cell therapy is most commonly to replace cells lost to the disease process. Although it's important to remember that cells can also be used to deliver a wide range of molecules to the brain. As you can see in this diagram, the principal cells that are lost as the disease progresses are medium spiny projection neurons that make up 80 to 90% of striatal neurons, with sparing of other neuronal subtypes. However, it's become increasingly apparent that glia are also heavily affected early in the disease process, and that glial replacement may also be a strategy for reducing striatal degeneration. There's a long history going back several decades of using donor cells derived from the developing fetal striatum. The value of these donor cells is that they've been through the process of normal development, so they're properly specified to become bona fide striatal cells. Use of these cells as a donor source gives us confidence in cell therapy as a strategy, as it's led to a large number of published studies demonstrating formation of functional synapses between donor cells and the host, and amelioration of functional deficits following transplantation of fetal cells into HD animal models. And there are also 2 pilot human studies showing functional improvements post-transplantation. However, human fetal cells are scarce and very difficult to work with, so it will be almost certainly not possible to do well-powered human studies using fetal cells. And so there's an imperative to generate cells from renewable sources, such as human pluripotent stem cells. Over the last couple of decades, there's been considerable effort from multiple groups worldwide to generate donor cells from pluripotent stem cell sources. I don't have time to review the science of this today, so I've highlighted work from members of the SC4HD steering group, which represents programs of work focused on delivering human ES cell-derived medium spiny neurons, human neural stem cell grafts, and grafts of glial progenitors. All of these programs of work being at various stages of readiness for future clinical application. It's important to say that this is in no way exhaustive and that there are other groups within the consortium with clinically relevant programs of work. Human cell therapy studies are highly complex with many issues that will require detailed consideration. For example, cell therapies require direct delivery of cells to the brain through a neurosurgical intervention. And there are also many other constraints that can impact on clinical trial design, such as the many months it can take for transplanted cells to mature post-transplantation. Therefore, with several substantial programs of work heading towards the clinic, we came to the conclusion that it was important and timely to start combining expertise to address some of these issues. So we established 2 related consortia: Stem Cells for Huntington's, SC4HD, focusing on cell therapies for Huntington's, and the EHDN Advanced Therapies Working Group, which considers a breadth of interventions that require delivery to the brain in Huntington's disease. To date, SC4HD has 28 members from 10 countries across 3 continents. We had our first face-to-face meeting in UC Irvine in 2018, hosted by Professor Leslie Thompson, and a second meeting jointly with the Advanced Therapies Working Group in Barcelona in 2019, hosted by Professor Josep Canals. Much of the last couple of years have been devoted to thinking and publishing on various aspects of the clinical pathway, with a major focus being to produce a position paper that has identified 7 key areas that need to be addressed in order to optimise clinical translation of cell therapies in Huntington's disease. These are defining principles to guide when a cell product is ready for consideration for clinical translation, issues related to the safe manufacture and scale-up of a cell product, guidance on the use of large animal models, optimal delivery of cells to the brain, especially considering the lack of a current CE mark device for this purpose, and a number of clinical issues, including ensuring efficient trial design whilst conforming to existing regulatory standards, establishing a framework for selecting patients and developing core outcome measures, considering strategies for optimizing graft integration and function, and importantly, considering strategies for reducing graft rejection. The next step is to establish task forces to work on each of these areas and to prioritise themes within each area. And this will be the focus of a virtual meeting of this group to be held on the 1st and the 4th of next month. In fact, one such task force has already been established and is being led by Professors Liam Gray from the UK and Romina Aram-Badin from France to consider challenges of therapeutic delivery to the CNS, starting with devices for delivering cells to the brain. So I'd like to finish by thanking steering committee and members of SE4HD and the Advanced Therapies Working Group for their work over the last 2 years, and also to thank our public and patient representatives, and finally to thank the CHDI Foundation, CIRM and EHDN for generous support of the face-to-face meetings. Thank you.